Hi, I'm Jonah Comstock, Editor-in-Chief at pharmaphorum. I'm joined today by Phil Johnson, the CEO of Interius Bioscience. Phil, welcome to the show.
Well, thanks, Jonah. Appreciate you taking the time to talk to us.
So Interius is in the sort of very exciting next gen of the cell therapy space, working on in vivo. So I'd love to hear a little bit about the company, how you guys got started, and what exactly you do. And then maybe we can talk a little more broadly about the space that you're in.
So thanks, Jonah. Interius is an example. You've heard often of this concept of bench to bedside. Everybody uses it. It's been around a long time. Interius is an example of bedside to bench to bedside. So we were founded by Dr. Sar Gill, who is an immuno-oncologist at the University of Pennsylvania. And Sar uses CAR T cells for his patients to treat his patients. And in the sort of peri-pandemic era, I call it, he, became frustrated with the length of time it was taking to get CAR T cells to his patients. Lots of manufacturing issues, lots of delays in getting the product to the patient. And so his basic concept was, hey, look, let's skip all the ex vivo manipulations that are required, including taking cells out of the patient, taking them to a manufacturing facility, turning them into CAR cells, reinfusing them. Let's just do it all directly in vivo. And so what that means is that instead of having to take cells out of the patient and go through all the steps, we're just injecting a vector directly into the patient that creates the CAR T cells inside the body, right where they live. And so hence the sort of the phrase that you used, in vivo CAR T. It's just another way of manufacturing CAR T cells, but in fact, directly inside the body instead of outside. So ex vivo versus in vivo.
You make it sound like it's much simpler, but I get the impression it's actually much more complicated than the ex vivo work that kind of dominates the space right now.
Well, actually, I would suggest to you that you're right. It is simpler, simply because all we have to do is manufacture the vector that makes the cells. We don't have to manufacture cells. We don't have to actually take cells out of the patient. And importantly, there's another piece of this that's really a differentiating factor is we don't have to treat the patients with chemotherapy before we infuse the vector. When you do ex vivo of any sort, whether it's, we'll use some jargon here, allogenic or autologous, you have to immunosuppress the patient so that the cells can actually engraft. In our case, since we're not engrafting cells, we don't have to do that. So the patients no longer have to have chemotherapy, which has a lot of side effects in and of itself. And this has been an extremely popular aspect of what we're doing with our key opinion leaders and our clinical advisory board.
Tell me more about this vector. What exactly is it that's going into the body and modifying these cells?
Yeah, so we're using the same basic vector backbone that's used to manufacture the cells ex vivo, but what we have done is engineered it specifically for use in vivo. So that took us a while. That was a couple of years long process to re-engineer the vector so that it could be used directly inside the body. And what we've done is done some fancy engineering to allow it to target very specifically certain cells inside the body so that it doesn't go everywhere. It only goes to certain specific cells. And these are the cells that we want to turn into CAR T cells. And in our case, what we've done is taken it actually one little step further, and that is that our vector targets both T cells and NK cells. And these are cells of the immune system, obviously, that can participate in eliminating tumors. And so it's really something that distinguishes us from, I think, anybody else in the field at this point is that we're creating both T and NK cells that can fight tumors.
And then what happens to the vector when it's done its job? How does it know when to stop?
This is in the jargon a non-replicating vector, so it doesn't replicate. It's simply a DNA delivery vehicle. So when it's done its job, it's gone. It goes away completely. And so this is not something like you would put a vector inside the body like, say, a measles vaccine that would replicate. This is simply something that targets certain cells, puts the CAR in the place in the chromosome of the cell, and then it goes away. It has no further function.
So talk to me about how far along you are. I know you guys have been around for a while now. How, what's your pipeline like? How many indications have you made this work for, and how close are you to the clinic?
The interesting part about this particular discussion we're having is we are now actively enrolling patients as of today into our phase 1 trial for B-cell malignancies. So we have two programs. The first is, as I just said, B cell malignancies. This is a program we call 2104. It targets T cells and NK cells, as I said, to turn them into cells that will kill B cell malignancies. That's program one. It's now in the clinical stage. So we're now, in fact, a clinical stage company. We have a second program that we internally call 2106, which uses exactly the same platform. We just tweak it a little bit so that transduces a different, it, it transduces the same cells, but attacks a different molecule on the surface of B cells. And this program is designed to treat patients with autoimmune diseases where pathogenic plasmablasts are the evil actor. They're making the antibodies that attack self, if you will. So two programs, one for B cell malignancies, the other for the autoimmune space. The second program is roughly a year behind the first program, but we are moving along very rapidly to get that program into the clinic.
What's so interesting to me about this is when you go to a conference on cell therapy, you talk to sort of the experts. It feels like the space is moving sort of so fast that we already have kind of like steps ahead of steps ahead, right? Everyone's talking about moving from autogeneic or auto from autologous to allogeneic, from having to take the patient's own cells out, modify them, and put them back in to using donor cells. And in vivo to me feels like it almost leapfrogs that. This is like the next step after that, but we're already working on it, already developing it. So how do you see the space kind of evolving? If everything goes well for you, do you see kind of people changing gears, or are there going to be some cases where in vivo is just a non-starter? Or
Yeah, so I am old enough and to have seen the dawn of the whole CAR T era, and I will tell you without hesitation that the inventions and the implementation of CAR T has been a revolution in treatment of hematologic malignancies. I think that nobody would deny that. There are patients now that are alive because of the work that was done at Penn and many other places back, 15 years ago. And again, you now have 6 approved products in that space that are on the market that are today treating patients and saving lives. We all recognize, however, that they, as we said earlier, it takes a while to make these products. They're also very expensive. And then the question is there a way to make them better? And so the mantra that we use inside Interius all the time is faster, better, cheaper. And so the faster part is, I think, pretty self-evident. We can call the pharmacy, have the vial sent down, and treat the patient on the same day. So there's no waiting around. The cheaper part is also interesting because we know that these products, the approved products, cost hundreds of thousands of dollars to make and administer. There's lots of technical fees associated with hospitalizations and so on. We now have calculated that we can actually make a dose for a patient under $10,000. And it's probably going to be less than that, depending on, of course, how the clinical trials go. So the faster and cheaper part, I think, is really self-evident. But the better part is what we're doing the clinical trials to prove. And the reason we think it has a chance to be even better is because we're not taking the cells out of the body like we talked about. We're not exhausting those cells by putting them through a lot of culture in bags and manufacturing facilities. These cells are in situ, in place, ready to go, and are not exhausted and are more stem-like, which is a phrase that we use all the time. It means that they're more native, they're more like themselves. And so the chance that those cells are going to actually be better cancer fighters, of course, remains to be seen, but it's a theoretical possibility. So faster, better, cheaper is something that we talk about all the time.
and you can add on top of that better patient experience, right?
Completely, yeah, and that's a huge point, Jonah. The issue of accessibility is huge, and so there are lots of data out there suggesting that less than 30% of patients who are eligible for CAR T therapies get them for a variety of reasons. And an overused term, but we'll use it here, is democratization. So we think this is a democratization of CAR type therapies for patients that don't have to be treated at ivory tower hospitals. They can be treated in community cancer centers.
One of the interesting things about the CAR T, the cell therapy space as it's evolving, is that there's this real sense of the infrastructures, like building a plane while we're taking off, and people are trying to see all the things you need in order to administer these very complex bespoke therapies, get them into not just the academic medical centers, but rural, so that everyone has care. And it's a huge project that like, and I guess that's the thing, like that's why it's so interesting to me that we're sort of already talking about in vivo, because it has the potential to almost obsolete that huge project that is only now getting underway. And I'm just, I mean, what will that look like? Or again, is this something where like there's going to be good use cases for it, and there's going to be things that are too complicated for a long time that we still have to do ex vivo?
Yeah, I think again, I don't think there's a line in the sand where you cross and all of a sudden things change overnight. It goes from dark to light. I think you'll see a gradual implementation of newer forms of therapeutics, whether it's in vivo CAR Ts or bispecifics or other modalities. The greatest news of all here, Jonah, is that patients will have therapeutic options available to them that previously weren't available. So you have patients, for example, who are not eligible for ex vivo CAR Ts for one reason or another, either cost or time, and they will be able to use in vivo therapeutics as opposed to having to wait for ex vivo cells to be manufactured. The whole concept of no lymphodepletion, no chemotherapy is, I think, a really, really big part of this. And patients who, have exhausted their options and don't really want any more chemotherapy will be looking for options like in vivo. So again, to me as a physician, it really speaks to having therapeutic options for patients that may not have other options.
How many people are working on this right now? It seems, pretty cutting edge. It's certainly, I don't think it's the first time I've heard of it, but people kind of tend to talk about it as something that's quite far off. Whereas you guys are already in phase one. I mean, obviously, getting to phase three does take a few years, but like, it seems well underway. Are you starting to see more competition, or?
Yeah. I think when we got started in 2021, there were a handful of companies that we knew about who were in the space that were either already there or were about to enter the space for in vivo. Because, as you have said, very astutely, this is the next generation. This is the next cool thing, if you will. And so I think as time has gone on the past few years, more and more companies and biotechs and academics have begun to realize that this really is the future and have begun to apply their own technologies to the space. So yeah, it's a growing group of companies that are doing this. We will be first in phase one trial, but there are lots of people lining up to enter the space as well. And so again, to me, that's great news because it validates our approach. It also gives again those therapeutic options for patients.
Yeah, and I know pharma folks, especially from a practice, from a clinical background like you, don't always like to talk about the things in like horse race terms, right? Where who's going to get there first, first market, because it really is about the patients. But yeah, in a space like this, I know there is a lot of pressure to sort of be the first one who proves it out, because and then for Interius, I guess, what comes next if everything goes well? Are you guys looking for an exit, or are you really hoping to kind of establish yourselves as a major ongoing player in this evolving space?
Yeah, more the latter, Jonah. I think our focus is really, really laser on getting clinical data for this approach, for our vector. I think once that happens, then the panacea of things that have become available will change, and again, offering patients options for therapeutic interventions that they did not have before. We will again proceed into the next year, gathering data on 2104. We'll begin to submit regulatory documents and enter the clinic for 2106, which is the autoimmune program. And beyond that, the really cool thing about our vector is that we can target almost any cell in the human body. So we can begin to think about other applications for our platform beyond just either hematologic malignancies or autoimmune diseases. So we have the capability really of transforming in vivo gene therapy in general as we move forward and get proof of concept.
Yeah, I mean, that's exactly what I was going to ask next. Is the other thing that I hear about cell therapy is that blood cancers and autoimmune are really just the start. There's a lot of people working on how can this technology be applied to solid tumor. There's people talking about other diseases outside cancer. So I mean, it, is there anything about in vivo that makes it different than than other kinds of cell therapy in terms of what's possible here, and the sky being the limit, as it were?
Well, again, remember, and again, this is something we've had to deal with, for a while now. We are not a cell therapy company. That's not what we do. We don't make cells. We actually make vectors that treat cells inside the body. So again, if I have a payload I want to deliver to, let's say, the liver or the retina or the lung, I can begin to modify and engineer our vector to treat those cells, not just T cells or NK cells. So again, it's a very, very broadly applicable platform that is, and we use this term sort of loosely, but it's a plug and play situation where we can switch out components and really address new applications pretty quickly.
Yeah, so you really are kind of building for the future beyond these first two candidates.
Absolutely.
That's great. Any other thoughts about your company or about the space more broadly before we wrap up?
Well, again, appreciate your time, Jonah. We are excited about getting into phase 1 trials now to show the world that this concept actually does work. It's been around for a while. We have, again, engineered a unique approach to this that we think has tremendous potential for patients and their families, and are anxious to really get moving on this as we are. And so by the end of this year, we will have treated a number of patients, and hopefully by mid next year, we'll have data that we can report out.
That's great. Well, I'm so glad to make the connection and learn a little bit about what you're doing. It's really interesting and really exciting. I think kind of potentially transformative work. So hopefully we'll keep in touch.
Thank you, Jonah. We agree with you completely.
That concludes this episode of the pharmaphorum podcast. You can find more information about this episode, including a download link and information about other installments in the series, at pharmaphorum.com/podcast. The pharmaphorum podcast is also available on iTunes, Spotify, Acast, Stitcher, and Podme, where you can find and subscribe by searching for pharmaphorum. And don't forget to visit our website, where you can sign up for daily news and analysis bulletins, and to follow us on Twitter at pharmaphorum. Thanks for listening.